So my name is Nayab Gill, and as mentioned, I am on a team of undergraduate students on campus doing research in the field of synthetic biology. We are competing in a synthetic biology competition called iGEM. iGEM stands for International Genetically Engineered Machines. So iGEM's goal is to promote research in the field. So every year, students from around the world get together with advisors from their universities, and they try to come up with new and innovative ways to use synthetic biology to make some new improvements in every field imaginable. So today's goal is to explain to you guys what synthetic biology is. Originally, this was designed as an activity for high school students to introduce them to synthetic biology, some of the principles of synthetic biology, and how it's actually carried out. So have any of you ever heard of the term before, synthetic biology? No? Okay. So about a month ago, I'd never heard of it before either. So when I got into the research group that I'm in right now, I got in as a human practices member. And it was really interesting to learn about all the possible things that can be accomplished using some of the principles given in synthetic biology. So before we get into what it is, how it works, how we carry research out using it, I think it's good to go over some basic foundations in molecular biology. So I'm thinking we'll start off with DNA. So what can you guys tell me about DNA already? Like, what do you already know? Okay.
Bases, it's got alternating phosphate and sugar backbone.
Right.
Held together by hydrogen bonds.
Okay. Anyone else? Okay, so you hit all of the right structural points. But DNA is the structure that carries all of your hereditary information. So all of your traits that are found in every organism, in humans and animals, everything, it's carried in your DNA. So as you mentioned, it's a ladder that's been twisted to create a helix. And the building blocks for DNA are called nucleotides. Each nucleotide has three basic parts. One is a phosphate group, the other one is a sugar group, and then the last one is a nitrogen base. And there's 4 different nitrogen bases that can be used. So as shown here, they are adenine, thymine, guanine, and cytosine. So when you look at the structure right here, the backbone of the DNA is created with sugar and phosphate reacting with each other, and then the rungs of the ladder are created by the nitrogen bases interacting with one another. So adenine and thymine like to interact with each other, and guanine and cytosine bind with one another. So as you're reading down the DNA strand, the order that the nitrogen bases are placed in is going to determine which trait what trait that marks for. So each 3 nitrogen bases come together to make an amino acid, and a group of amino acids come together to make a protein. So proteins are the worker bees of all organisms. So if you want to create a blue eye and you have a DNA strand that codes for a blue iris, that's going to produce a protein that's going to create a blue iris. So any questions about that? Have we heard some of this before? Yeah, okay. So this is like a basic intro into how DNA goes to protein. So let's look at that in a little bit more detail. So before we go into that, your entire set of DNA sequences of all organisms, that's called your genome. And then one hereditary trait is going to be a gene. So a gene is a discrete set of DNA that codes for a certain protein or a group of proteins that carry out one function. So the process of going from DNA to protein is called the central dogma. So the central dogma can be broken down into 2 basic steps. So this is an extreme simplification of what's happening at the molecular level, and we'll need it to move on into how we use synthetic biology in the lab. So the first step is called transcription, and that's when you go from DNA to RNA. You have an enzyme called RNA polymerase, which is right here in the figure. And what it does is it unwinds the DNA and it makes a single-stranded copy of it. So RNA is essentially the same thing as DNA in a single-stranded form. The only other difference between it is that RNA polymerase replaces the thiamine with a uracil. So instead of having an ATGC, in RNA you have an AUGC, but it carries the exact same information. So now you have an RNA template. So the next step is translation. In translation, you have a ribosome, which is right here. It goes along this RNA strand and it reads all of the nitrogen bases. As I mentioned before, every 3 nitrogen base makes a amino acid. Okay, and then a group of amino acids come together to make these proteins. So in translation, you have a ribosome. It's going to read the RNA and it signals transfer RNAs to come along and transfer amino acids to the cytoplasm where this is all taking place, and then you get the formation of a protein right here. So are we clear on how all of that works? Are there any questions? Yes?
On one DNA strand, there will be many alkyne bases
Okay.
in just a line in order, and when that be translated to RNA, the same thing. There will be many those alkyne bases to be used for protein synthesis or to select what kind of amino acids. So I just wonder, maybe those enzymes knows to start from what?
Exactly. So we're
Alkyne
going
base.
when
But
we
human beings, how we know that? How we can know that? Do you have any clue?
how we can know that.
Yeah, because for example, in nature, those enzymes knows to start from what base to select one specific amino acid. But we human beings, how, do we know that? Start from what alkyne base?
Which base do we start
Yes.
off of?
Yes, which base do we start?
We start off with an AUG. It's a methionine amino acid.
Say it again.
You're asking where does the ribosome know to start
For
reading?
example, you have the ATCG, so many.
Right, okay, so when we move on into the next part of our presentation, it goes into more like there's a start codon that we use. So once we go on into the further steps of how we manage this in lab, we'll further explain that when we get there.
Okay, good.
Okay. All right, so what's synthetic biology? We just spoke about the central dogma, and synthetic biology uses the central dogma to its benefit to create or change systems that already exist in the body. So as stated here, it's the use of engineering principles to design or redesign a natural biological system through alterations of the system's genomes. So you're taking either something that already exists, modifying it in some way, and hoping that it will get carried out to what you want to see in the organism. So when I was first given this definition, I was a little confused about how does that happen? How do you modify a gene? So I like to look at it as a computer. So if you look at a computer, engineers like to use binary code. And to the untrained eye, that's just a sequence of ones and zeros. So I'm not sure what any of that means, but it's really sophisticated information that tells a computer to carry out very specific tasks. So over here, we're showing a binary code that's being input into the computer, and then the computer creates an output. And in this case, we decided to make it a binary sequence for the iGEM symbol. So organisms work in a similar fashion, but instead of using binary code, you're now using DNA sequences. So in this case, we have a bacteria, which is red in this image, and we're going to insert a sequence for a bacteria that's going to change its color to green. So once we put that in, we get an output similar to a computer of a bacteria that has a different color. So that's as far as computers go. The cool thing about using organisms is that they have the ability to replicate what you just created. So you not only have created something new, but your organism is going to replicate it for you on its own naturally. So synthetic biology has been used in agriculture and many other parts of our world for decades now. But we're only recently starting to realize the potential that can be reached using some of these principles. So let's talk about a little bit of success that we've already had using genetic modifications, using synthetic biology in agriculture, since you guys are here on behalf of NewBio to learn about a sustainable energy future. So the first genetically modified organism to ever hit the market was the flavor saver tomato. So what scientists were able to do was they were isolating a gene that made the tomato squishy. So they suppressed that gene, which increased the tomato's shelf life. Another thing that has been done is creating BT corn. And I'm sure some of you have heard about BT corn before. What they did was they isolated a gene that was only toxic to caterpillars. So when caterpillars ate the corn, they died. But when humans consume it, it doesn't harm them in any way. So this decreased the need for fertilizer when growing corn. Another thing that I'm sorry you can't really see it very clearly, but they've been used as synthetic biology has been used to create nutritional supplements and vaccinations. So golden rice was created to help fight against vitamin A deficiencies. It's rich in beta carotenes, so when consumed, your body stores all of it, and when necessary, it's converted into vitamin A. Another thing that's been created is a potato that is vaccinated with cholera. So cholera is an intestinal disease that occurs when you eat food that's not really sanitary or water that's not very clean or pure. So this is where synthetic biology has been used successfully before. But we're seeing all of these new technologies coming out that are able to push this even further. So I'd like to give you a little bit of time to kind of talk about where you think it can be applied using NewBio in agriculture or in any other field that you'd like to. But think about some applications of how you can use genetic modifications and things like that to make something better.
Offspring.
Right, so it's going to replicate.
Okay.
Yeah.
Okay.
All right, so let's hear what everyone came up with. Would someone like to share? Anyone?
This has already been done, but there was an amino acid that promoted wool growth in sheep.
Okay.
They found that amino acid in a gene sequence in the chicken. So they took it out of the chicken and they put it into alfalfa hay. And then they fed that hay to the lambs. The lamb actually grew more wool.
Well, that's amazing.
So it was kind of interesting that it went from an animal to a plant and then back to an animal.
Okay, all right. Anything else? Okay.
We're
Yes.
actually kicking around the idea that could we make somehow introduce an enzyme that at dormancy would begin to break down the lignin more quickly. Because lignin is the challenge to reducing getting the carbohydrates back out to begin the manufacturing process of the biofuel.
Right. That is a great application towards NewBio. Go ahead.
Can we, in plants that don't fix their own nitrogen, somehow get them prepared to fix their own nitrogen, cross with some sort of legume or?
Mm-hmm. Right, that's really good as well. Anything else?
They said in some construction, in the south where they have termite issues, some kind of, in the wood that is more resistant over time that, to those insects.
Right.
Or even, I don't know, we didn't know much about the straw bale, the Montana house you were talking about, the straw bale construction, the insulation.
Okay.
I don't know how long that lasts in there, but, even improving that if there was a problem with that over a long period of time.
Okay.
I don't know what the shelf life is, literally, that Montana house about?
Okay, so you guys came up with really great specific, targeted improvements that you can make. So when we came up with potentials, we tried to come up with potentials that can be used across the board anywhere in the world. Of course, these can be targeted and used with NewBio's endeavor to a more sustainable bioenergy future. So you can increase crop efficiency using synthetic biology. So some of the ways to do that would be to create crops that use less water. In drought-ridden areas where water isn't such a everyday thing that we see it, where if plants are using less water but creating the same yield, that's very efficient for them, and it'll increase agriculture for those areas as well. You can, as you mentioned, creating plants that use nitrogen more efficiently and things like that can be improved. Creating plants that have a shortened growth cycle, that's what that says. I'm sorry again that it's not showing up here. But yeah, like shortening the growth cycle of a plant so you can have it on the market faster, but create the same yield, have the same product produced, but at a faster rate. The last thing that we have up here is to create plants that function the same under limiting conditions. And this is one thing that I think is very applicable to NewBio, because the goal is to grow switchgrasses in marginal agricultural and mine lands where resources aren't the same, they're very scarce. So the ability to have plants that function well under stress is a great thing to have. So the last thing that we have up here is to optimize production of desired products. So plants create a multitude of things that are used, like fruit, like smells, and biofuels, as you guys are focusing on. So having the ability to target in on enzymes that create those things and to overexpress them and have them create more of it is a great thing to have. iGEM this year is also working on plants, and 2 of our projects are doing things similar to the NewBio biofuels. So we're having a project that's creating butanol, and then we have another project that's creating the vanilla scent and vanilla flavor in plants. So it's already found in plants, but we're trying to overexpress those pathways and have them create more of it so we don't have to create these in a lab or outside using harmful chemicals and such. So right now I'm going to have one of our other